There are several differences between prokaryotes and eukaryotes during the process of DNA replication. And over here, I just want to highlight a few of them so you can have a little better understanding of the processes in the two different kinds of cells. So let's go ahead and look at prokaryotes. Firstly, it's important to remember that the prokaryotic DNA is a circular DNA. So here we have a circular DNA which is present in the prokaryotes, and the eukaryotic DNA is called as linear, which means that it's a straight line. You know, you have a straight, long DNA strand. Now, because the DNA in the prokaryotes are in a circular form, so a single circular loop of DNA must be replicated before the cell divides. So if you can imagine, if you have, for example, something in a ring format, if you have one ring or a circular, you know, a ring, a wedding ring, or some sort of bangles you can imagine, and before you can have it replicate, it actually should have, there should be a replication that should take place, and you will have two of these rings very close to each other and then separate out. Now, the origin of replication in a prokaryotic is at different stages. We call the origin as where the helicase will come and start replicating. So it happens at actually different regions, and it will grow in both directions. It will grow this way, it will grow this way. So eventually you will have a complete a process of replication which is actually going on in both directions. So it's a two-direction process of replication. And the process must again always be happening in the same 5 to 3 end. However, in the eukaryotic, because the DNA is so long, there are certain regions that are called as a replication fork, where it can take place over here, it can take place over here, and again it's happening in both directions, and eventually the replication forks will meet. This increases the frequency, or the rate of the reaction, I should say, not even the frequency, the rate of the reactions, because there are many origin of these replication forks. In prokaryotes, often you can have only one or two replication forks. You don't really have a whole lot, but it can happen in a more two-direction manner in both of them. So this is something you may want to see the shape of the DNA and how it sort of replicates. The other quick topic we want to cover in molecular genetics, which actually leads to the evolution, would not be possible without molecular genetics and without a process called as mutation. Mutation is a permanent change in the sequence of the bases. Some mutations are due to errors in the DNA process, and if there's an error in the DNA replicating process, there are enzymes that will come and proofread the sequences. So for example, if my first sequence is A, T, C, A, T, and then the complementary should be T, A, G, and for example, if this was missed and you have an A over here, that is, you've skipped a nucleotide, sometimes there are enzymes that will come and fix those changes, because it's going to hinder in the long run. And we'll talk about this later in some other topics of DNA expression during protein synthesis. But the cell actually has a proofreading process, a natural process which checks this incorrect replication process. Because if these are not corrected, then can lead to a disease such as cancer. Is also one of the diseases that can result due genetic incorrect correction of the strand. However, I want to keep in mind that mutations are not always harmful. Not every mutation is going to lead to cancer. Mutations also will lead to a change in the genetic makeup, which may be good and give variation in a population. So one of the examples of a DNA repair enzymes are thymine dimers. This is not from your book, but I found them from another source, where you can see a thymine dimer is a DNA polymerase. It recognizes that, and the polymerase recognizes and binds to the thymine dimer. And in this presence of visible light, the enzyme catalyzes the chemical cleavage of the dimer, therefore restoring the normal base pairing and repairing the DNA. A thymine dimer means that if you have thymine on one strand and also on another one, thymine actually has sulfur, and it binds up to give a new kind of bond. So just an example you may want to remember is a thymine dimer, which will help an incorrect mutation. So DNA repair enzymes are usually available within cells, and they're able to reverse most of the damage to DNA. But again, some may be skipped, and those skipped ones may either give you variation, for example, a variation in different kinds of proteins, or it can give you a genetic disease, for example, sickle cell, or it can cause more aggressive diseases such as cancer.